Okay, let's talk about our seedless vascular plants, which we just a lot of times call SVPs. So the seedless vascular plants have vascular tissue, aka xylem and phloem, and if you're not sure what that is, don't worry, we'll explain it in a second. But what this vascular tissue does, it's essentially like the veins in a plant, which you might be able to see in the leaf. Well, those veins also exist in the stem, and they allow for a much more efficient transport of water and nutrients from the ground up through and all around the plant. So this was really, really important to establishing independence from water, and it also allowed plants to become much larger. We are going to talk about two phyla. A little pen here. We're going to talk about two phyla, the Lycophyta and the Monilophyta. So here are our Lycophyta, also just called the lycophytes. And then here are our monilophytes, phylum Monilophyta, which includes the horsetails and the ferns. So if we look at our little cladogram here, and we want to see kind of the key synapomorphies, some key traits, whoops, not good, that unite our groups in the seedless vascular plants. Well, vascular tissue is probably the most important of them all. So what is this vascular tissue stuff I'm talking about? Okay. All right, so the vascular tissue is capable of efficiently transporting materials over long distances in the plant. And essentially what it is, there are two types. There's what we call xylem. So xylem are cells that are specialized to move water and the nutrients that tend to hitch a ride along with the water. So the main type of xylem cells are called tracheids. And these tracheids tend to be very hard, and they are hardened by a polymer called lignin, which is the stuff that makes wood hard in a tree. So not only does it do a great job at transporting water around, but it also makes the stem and roots harder, stronger, faster, better, all that stuff. Let's find a picture that is... Okay, so this... let me just start over. I wanted to find a picture. So this is like a cross-section through a stem, and let me get back to my pointer. So here's our cross-section of a stem, and so each of these holes you can see here are the tubes of the xylem that would conduct water. So here's kind of a long section view, I guess, to kind of show you the idea that each of these circles you were seeing in that cross-section is actually a tube. And this tube goes all the way from the roots up through the stem, through the leaves. And again, you have probably observed this by observing the fact that leaves have veins. Those veins are, among other things, the tubes of the xylem. So xylem, very important for moving water around. And typically strengthened with something called lignin that makes it very, very tough and hard. And these are one-way tubes. They go typically from the roots up through the plant, and then out through the water vapor can be evaporated out through the stomata. Phloem is the other kind of vascular tissue. Phloem are cells specialized to move sugars. and, other metabolites such as amino acids or organic products. But their primary job is to take sugars from wherever they are produced, such as in the leaves, and to transport them to where they are needed, maybe to the roots for storage, or maybe to a developing fruit or flower that needs the sugar. So these are two-way pipes. So essentially this xylem and phloem is a series of pipes through the plant that move around water and move around the products of photosynthesis. So this was a really important adaptation to help make plants more independent of water, because they could now get it from the ground. So this vascularization, this presence of vascular tissue, was important not just because it allowed them to get water from the ground, it also allowed them to get much taller. So particularly the xylem is very hard and tough and would allow the plant to actually be able to get tall without sort of just kind of flopping over. So that was kind of a really big deal, because plants need to intercept as much sunlight as possible. So getting taller, right, if we have plants at ground level, well, if you're able to get taller, you can intercept more light than anybody else. So this actually led to the evolution of taller and taller plants, because they were competing for access to the sunlight. So some other characteristics of seedless vascular plants, a branched, so we see some branching going on, a branched diploid sporophyte that is independent of the gametophyte. And one big deal is that the sporophyte is the dominant generation. So it is the larger, more conspicuous plant. So think of something like a fern. The thing that you would think of as being the fern, that is the sporophyte generation. And the gametophyte generation is quite small, maybe the size of like half of a dime or something. So, again, the other thing we got was more branching. And branching was a really good adaptation because it allowed, basically it gave us more areas to stick a sporangium, so that we could make more spores. We also saw true leaves and true roots for the first time in our seedless vascular plants. So leaves, so let's think for a second about what a leaf is. It's basically a flattened portion of the plant, and its whole job is to increase the surface area to perform photosynthesis over, right? It's like a solar panel, right? We make those flat and large so they can intercept more sun. So here you're seeing a couple of images of sort of the stages, the hypothesized stages in the evolution of leaves. And there's two basic leaf types we run into in our seedless vascular plants. There are what we are called microphylls. And we see this in our lycophytes, in our phylum Lycophyta. And what a microphyll is a small, spine-shaped leaf that is supported by a single vein. And the vein is vascular tissue. And the hypothesis of how these perhaps came to be was that a sporangium started to evolve into a more leaf-like structure in order to, sort of straightening out, flattening out, in order to intercept more sunlight so it could play a greater photosynthetic role. Now, we also have a more classic, what we would think of as leaves, are actually known as megaphylls. So we see these in our monilophytes, our ferns and our horsetails. So these leaves are larger, and they're pretty diverse in form, and have a highly branched vascular system. So unless you spend time, at either a place that sells plants or a greenhouse, I can pretty much guarantee every leaf you've ever seen is a megaphyll, because you're just not super likely to have run into a lot of club mosses. You might have, though. You might have. But most leaves, most of the things you would think of as leaves, are megaphylls. And sort of the idea of how megaphylls came to be was that in a more highly branched plant, some stems started to become progressively reduced and flattened, and photosynthetic tissue started developing between them. And of course, this is selected for because you are doing more photosynthesis if you have this nice flat surface, photosynthetic surface, to intercept more sunlight. All right, so let's talk about some of our seedless vascular plant lineages. Starting with the phylum Lycophyta. Okay, so the phylum Lycophyta includes three major groups: the club mosses, the spike mosses, and the quillworts. There's about 1,200 species that are, currently extant, which means not extinct. And these are relics of a much greater past diversity. Current species are all small, but that didn't used to be the case. In the Carboniferous period, lycophytes actually got to tree size, big giant trees that could be 2 meters in diameter and over 40 meters tall. So they were gigantic. So most members of the phylum Lycophyta have what are called strobili, which are clusters of spore-bearing microphylls at the tip of a stem. What's a sporophyll? So, sorry, we would call these, we would refer to these as sporophylls. Hyll is a leaf that is okay. Let's take a look at some examples. So here is a strobili, which is essentially a cluster of sporophylls. And a sporophyll is basically a microphyll that has that is specialized to house a sporangia. So they're really nice and apparent here. We don't see any apparent ones here. These ones are much harder to see, but we will look at some spike mosses, and you will actually be able to see that at the tips of some of these branches, we have some of these strobili at the tips. Okay. All right, so let's get to our next phylum, Monilophyta, which is the ferns, and these are the ferns here, and the horsetails over here. So talking about the horsetails first. They were once quite diverse at one point in time. Now most are extinct, and there's just one genus that exists with only 15 species. So these form a strobolus, oftentimes a terminal strobolus, a really nice apparent one you can see at the tip of this horsetail. And they have this kind of interesting jointed growth pattern where you can see these really apparent joints. And there's some of these growing just outside of the H building, in between the H building and the D building, so you can actually look at these. And what happens is that we have these rings at these joints. There are rings of leaves and sometimes branches. And these leaves, although they're teeny tiny, they're not microfills. What they are highly reduced megapills. So it's really common to say, wait a minute, I thought you said Monilophyta had megapills. These are microfills. Nope, they actually are highly reduced megapills. But obviously they're not going to do much photosynthetic work, so the stem is the main site of photosynthesis. Another kind of cool thing about the horsetails, if you touch one, or if you ever have, it feels very rough to the touch, almost like you could cut yourself on them. And that's because the cell walls of these plants have silica deposits, so that's the main component in most glass. And so they have this durability, and it makes them very rough. And probably, from the plant's perspective, it's great for deterring predators. Sometimes these have the common name scouring rushes, and that comes from the fact that you can actually use some species, the fact that they're so rough, they could actually be used to like scour a pan. So that apparently is something that people did back in the day. I guess make sure it's not a poisonous one, but sounds interesting to me. All right, so let's also talk about ferns. So let's talk about ferns. All right, so ferns have megaphylls, and we call these megaphylls fronds. They have what we call a sort of immature frond is known as a fiddlehead. I'm sure you've seen a picture, right? So it kind of comes like this, and then this fiddlehead will sort of slowly unfurl into the classic fern leaf. And of course, the actual appearance varies species to species. But the interesting thing is where we're making the spores. So the fern sporophyte, which of course is the larger dominant part of the life cycle, it's what we would think of as a fern, creates spores on the underside of fronds. So some of the fronds are what we would call fertile fronds, and if you flip them over, which you can see right here, you see these little brown clusters. Well, if we were to zoom in on them, we would see that they are actually clusters of sporangia. So these are called sori. These little clusters are called sori, clusters of sporangia. That produce numerous spores. So if we were to zoom in on one of these, we might see that it looks something like this. So here's a sori, and we can see we've got at least 1, 2, 3, 4, 5, 6, 7, probably much more, 8, 9, several of these sporangia at different stages of maturation, and each of these sporangia would hold numerous spores. In this case, we have a little extra flap of tissue called an indusium that provides an extra layer of protection to these sporangium. Some species of ferns have an indusium, some don't. It's actually a character we use to help identify ferns. So looking at the fern life cycle, what we would call the classic fern that we all think of is the sporophyte, and it is going to house the sori, the clusters of sporangium, under fertile fronds. So not every frond has these. If it's a sterile frond, it might just have photosynthetic jobs and no fertility, but some will have these on the underside. So one sporangium looks a little something like this. And so these sporangium have cells that will undergo meiosis to make spores. And these spores will germinate into the haploid gametophyte generation. The gametophyte, so here's a fern gametophyte, and we'll look at one in lab. They're quite small. They're maybe the size of like a small pencil eraser, maybe. They're pretty little. And it's basically just one cell thick. They have some rhizoids for anchoring. And then we also have the gametangia. So this gametophyte has both male and female gametangia on it. So up close to this notch here is where we have the archegonium producing eggs. Down here we have the antheridium producing sperm. Water is required. So we're not completely independent from water here, because a drop of water, at least a drop, is needed for these sperm that have flagella to swim to the egg in the archegonium. And once that happens, fertilization occurs, we get a zygote and then an embryo, and then we have the sporophyte growing out of the gametophyte. But in this case, this, maybe at its very early stages, this sporophyte might derive some nutrition from the gametophyte, but very, very quickly, this sporophyte's going to get much larger than the gametophyte and be perfectly independent. So let's look at a couple different types of sporophylls in our seedless vascular plant. Again, sporophyll spore leaf. That's what it means. This is the leaf that has the spore on it. So basically, a sporophyll is a leaf modified to bear sporangia. And that sporangia is our spore house. It's what's holding our spores. So fern sporophylls just look like normal leaves, but they have those sori on the other side, those little clusters of sporangia that hold spores. Lycophytes have their sporophylls that are modified into this cone-like structure we call a strobilus. Okay? Remember that strobilus I talked about? So this is a sort of up-close view. And what we can see is what appears to be like microphylls, but each of them is bearing a sporangia. So a cluster of these sporophylls together at this terminal, tip here, we would call that a strobili. Okay, so let's talk about spore formation in seedless vascular plants, because there's a couple different ways this can happen. In seedless vascular plants, you have a life cycle that is either homosporous or heterosporous. In a homosporous life cycle, there is one type of sporangium that produces one type of spore, and this spore will develop into a bisexual gametophyte. And what I mean by that is it's going to be capable of producing both sperm and egg. The other type of life cycle is when you're heterosporous. So if you are heterosporous, it means that there are two types of sporangia. that produce two types of spores. And let me talk about those two types of spores. There's a megaspore. It can make a megaspore, which is produced in a megasporangium. And it will develop into a female gametophyte. So this female gametophyte will be capable of producing eggs only. Let me just... I added on here two types of gametophytes, right? If you're heterosporous, you have two types of sporangia that produce two types of spores, each which yields a different type of gametophyte, male or female. So one type of spore is the megaspore that is produced in a megasporangium and will develop it into a female gametophyte, which of course will produce eggs. The other type of spore is a microspore. A microspore is produced in a microsporangium and it will develop into a male gametophyte. Which of course will produce sperm. Okay, so I know this is a lot on one slide here, but I did want to have them sort of side by side for comparison purposes. So this is an example of a homosporous life cycle, and the moss would be an example of a homosporous life cycle. So with the fern, actually everything we've looked at so far for plant life cycles that we've shown an actual life cycle for is homosporous. So we have a spore that germinates into a single type of gametophyte. And this gametophyte is capable of producing both archegonium and theridium that will produce sperm and egg and fuse in fertilization, giving us a zygote, and so on and so forth until we make spores again. Here, basically, it's a little more complicated in the heterosporous life cycle. So in this case, let's start with this sporophyte, okay? In this case, our adult sporophyte produces two types of sporangium, a megasporangium and a microsporangium. So they don't just make one type of sporangium, they make two types. The megasporangium produces megaspores, and the microsporangium produces microspores. The megaspores will germinate into a megagametophyte, which is female only and can only produce eggs. The microspore will develop into a microgametophyte, which is male only and can only produce sperm. And then it's all the same. Sperm and egg unite in fertilization, and we get a zygote again.